Our next presenter is Dr. Keith Perry. Keith received his bachelor's degree from University of California, Berkeley, and a master's in plant pathology from UC Davis, and a PhD in plant pathology from Cornell University. And he served as assistant associate professor in the Department of Botany and Plant Pathology at Purdue University before rejoining Cornell University in Ithaca as associate professor. And currently he's an endowed professor of plant pathology and director of the New York State Foundation seed potato program. Keith, thank you very much for being with us today. His presentation is Detection and Genetic Diversity of the Virus.
Very good. Glad to be here. I presume the sound is working?
Your sound sounds good. Just go into presenter mode and you should be all set.
Okay. So it's my pleasure to participate in the webinar. I thank you all for the invitation and wanted to describe some of our work on detection and genetic diversity of the red blotch virus. And I thought I'd start out, I'm working at Cornell University in Ithaca, New York, and I thought I'd start out by acknowledging the two cooperators in this work. Let's see, I'm having trouble with, okay, Jeremy Thompson in working with me here in Ithaca, and Mark Fuchs up in Geneva. We're all in the same department. So the three topics I was going to mention, I'll begin with talking about virus detection, have a few comments on genetic diversity, and then we'll finish up with a brief description of detection and diversity in wild vines that we've observed recently. With regards to detection, I thought I'd begin with visual symptoms and add a cautionary note that although there may be some very typical or classical red blotch symptoms as shown in these slides here, that they can be misleading. So I'm comparing here the leaf roll symptoms on the left side in each of the two photos, and red blotch on the right side. The left panel has the upper surface, right panel the lower surface. That's reversed. And these symptoms, while very typical, there can be gradations in between the two, at least what we've seen on the East Coast, and so any visual assessment needs to be followed up with laboratory testing to confirm it. For virus detection, PCR for the detection of the virus DNA is the only option we currently have available. There are no serological assays such as ELISA. And because of the caveat of PCR detection requiring a perfect match with the primers used and the virus, and you could potentially miss variants, we also make use of rolling circle amplification in combination with the PCR work that we do. This slide here shows the results from a PCR assay, a multiplex PCR assay. It's an agarose gel stain with the DNA products of the PCR assay. The stain gel bands are virus specific. The primers target the coat protein and the replicase genes of the virus. We have an internal control to help us make sure that the PCR reaction is working. And shown here are the products from extracts of red blotch infected plants and uninfected plant control. That was a multiplex PCR reaction, another that we mostly rely on. This is an example of a simplex PCR assay, one of the first developed, where you get a single band. In this case, the primers are targeting the V2 gene of the virus. And this is bringing what I just showed you together and showing you the positions on the genome where the primers bind, the PCR products arising from the primer shown in blue. There's two of them, one for the rep protein, one for the capsid protein, and the second simplex reaction gives you a product corresponding to the V2 gene. So a few additional considerations about the detection of red blotch by PCR. We have not observed any isolates for which the multiplex assay has failed to detect the virus, but both primer pairs do not always give rise to products. Sometimes we miss seeing the coat protein specific gene product. In our hands, the multiplex assay is likely more reliable than the simplex assay, so we have stuck with that. There are real-time PCR assays in use that are used by commercial labs and other diagnostic laboratories, so both conventional and real-time PCR is employed. And then an added note that some vines that have been confirmed as red blotch positive by sequencing have subsequently tested negative. I share this so that others are aware that this may occur. We infer that there is significant transient changes in the concentration of the virus, and this has been observed for vines both in the field and that we've returned to and those in the greenhouse, but we have not thoroughly investigated multiple seasons of the testing. And lastly, a rather unusual comment, but I think it's helpful for those doing diagnostic testing, that red blotch is the most difficult virus we have worked with in terms of detection, and that there's more beyond the more straightforward PCR gel results that are indicated in publications, that we've had many challenges along the way with inconsistencies, and while it works reliably now for us, it hasn't been straightforward. There are some anomalies in our understanding of red blotch and its detection. First of all, to my knowledge, no virus particles have been observed for the red blotch virus. There are no virus proteins that have been observed, such as the coat protein. With regards to antibodies, antibodies have been produced that recognize synthetic peptides and E. coli express coat protein, but no viral antigen has been detected in plants. And it could be a simple matter of the concentration of the virus in plants, but something is quite unusual about this virus. We've also transiently expressed the coat protein gene in leaves using an agrobacterium vector, and we just don't see any recognition or a stainable band. And we've pursued these analyses in westerns. So take-home message there is something is quite unusual about this virus. And lastly, there are in-field diagnostics for red blotch in development, and I suspect we'll be seeing at least one of these products in the coming year. With regards to genetic diversity for red blotch, first question is, why do we study genetic diversity? And basically, our main motivation has been a knowledge of the genetic diversity, and conserved viral sequences facilitates the design of reliable diagnostics and the production of clean planting stocks. It also provides us a genetic fingerprint by which isolates or strains of the virus can be tracked. And it's more difficult to do these kinds of comparisons of diversity if you have low, or fingerprinting rather, if you have low diversity in the field. And lastly, some strains of the virus may differ in virulence with genetic markers. And with genetic markers, these can be monitored. This latter case has not yet been observed. Our motivation presently is the first 2 points I brought up here. With regards to what we've seen, there are 23 isolates for which whole genome sequences are available for the virus. The isolates group into 2 genetic clades. Clade 1 has the fewest numbers of isolates. There's 95% sequence identity, or about 5% differences among those isolates. A lot more apparent differences in Clade 1 than in Clade 2, which are more homogeneous, and these members share 98% identity. Between the 2 clades, we have up to 8% sequence heterogeneity. Another way of looking at those 2 groups of viruses is shown here in an unrooted phylogenetic network, and I'll draw your attention to the left-hand side where you can see a number of linking lines, which is consistent with what we've observed in that there's greater extent of apparent recombination in this group, and by contrast, the isolates from the other group are a lot more homogeneous and clustered without this apparent recombination. And so if we were going to see strains emerging, it's more likely it might be from the first group, the clade 1. And the last comment on this genetic diversity with regards to selection in the virus, in that 5 of the 6 open reading frames are under strong purifying selection or negative selection. So even though we don't see these proteins, we know that we can infer that they're functional and being selected for within the host plants. And I'll finish up with a couple of comments about the detection of red blotch in wild vines. And this work began with an observation by Mark Fuchs about the distribution of red blotch affected vines in a commercial vineyard, and a schematic of that is shown on the slide here, where the red boxes indicate vines, 1 box per vine, in which had red blotch symptoms and where the presence of the virus was subsequently confirmed. And there was an unusual bias and clustering of the vines suggested a possible movement of the virus. So the context of this field is shown here. This was a field in Napa Valley. It is at one end of a field, and it's adjacent to a riparian woodland up at the top of the image on the right, along with a water source, a river, and within that woodland there were wild vines growing. So we proceeded to assess the genetic variability among vines within the affected field, and also tested for vines in wild plants in the riparian habitat shown at the top of the figure here. Each circle and number corresponds to an isolate of the red blotch virus, which we identified in those plants and then subsequently sequenced the genome of. So we sequenced the genomes for 3 of the 4 wild vines, from Cab Franc, 3 different clones, from a Merlot and a Cab Sauvignon clone. And to summarize this work, first we wanted to look at the variation, sequence variation, genetic variation within just one clone, within one plot. So we pulled out 44 isolates from Cab Franc clone 214. They were all clade 2 isolates, and they all were identical in sequence in a 587 nucleotide region. So within a given clone, there's no heterogeneity, no differences. 6 of the 7 cultivated vines contained clade 2 isolates, and these were sequenced. And among these isolates, these 6 isolates, there was limited sequence diversity, about 0.4%, which corresponds to about 13 changes within the genome as a whole. So at least between clones, we were able to see differences in the virus. Red blotch was detected in 4 wild vines, and 2 of these wild vines were typed as a hybrid of the native Vitis california and Vitis vinifera. And lastly, the genome of 1 wild vine isolate was nearly identical to that of an isolate from a proximal vine of Merlot clone 30, which that particular plot had been in the field about 30 years. It's since been pulled. So in conclusion, the PCR detection is presently the only option we have, and in our hands the multiplex assay is likely more reliable than the simplex assay. Red blotch is an atypical virus and in some cases can be difficult to detect. There are 2 genetic clades of the virus with up to 8% sequence heterogeneity, and no as of yet recognized biological differences among isolates. And lastly, red blotch has been detected in wild or free-living vines. And I'll close by acknowledging those who have done the work listed at the top of the page here, particularly Jeremy Thompson. All of this work was done in collaboration with Mark Fuchs in Geneva in the same department. A number of others have helped us along the way. Sudi with USDA, Foundation Plant Services, colleagues in Davis, commercial testing companies in California, and many others. And if I've neglected to list you, my apologies. Our funding has been from a diversity of sources: USDA competitive grants, the National Clean Plant Network, a number of the industry research funds from California, and also some from New York. And lastly, the California Department of Agriculture also contributed largely. And with that, I will close the talk.